Hi, my name is Hebah Khateeb. I'm a senior undergraduate student majoring in microbiology at East Tennessee State University. Today, I'll be presenting to you a project that I have for my honors thesis called or titled Analysis of Microglial Cell Activation and Morphology Change in an Animal Model of Treatment-Resistant Depression. So let's begin with some statistics about depression. About 1 in 5 Americans will have a major depressive disorder sometime in their life. At its worst, depression can lead to suicide, and close to 800,000 people die due to suicide every year. And so with more than about 300 million people affected, it's a huge burden to both on the individuals and society, with an economic burden costing over $210 billion per year just in the United States alone. And in fact, the World Health Organization actually recently decided depression to be the leading cause of global disability. So some clinical symptoms that you might have been aware of about depression include, anxiety, lower energy levels, disturbed sleep, insomnia, low pleasure in things, which is called anhedonia, and difficulty concentrating. And the rating scales of depression varies between mild, moderate, and severe depression. And severe depression, which mostly often results in suicide completion. And so when it comes to the standards of care, common antidepressants are the first line of treatments for moderate and severe depression. In the 1950s, the monoamine hypothesis of depression was formulated, which suggested that depression was associated with a deficiency in the transmission within the monoamine systems. The monoamine systems include like serotonin, norepinephrine, and dopamine, just to name a few. And from those days, so since the monoamine hypothesis was formulated, drugs were produced to be able to increase the brain concentration of serotonin and norepinephrine via different mechanisms in our nervous system. And unfortunately, the numbers are alarming when it comes to antidepressant use in the United States. It has become a multi-billion-dollar market, so about 11 to 13% of Americans aged 12 years and older are currently taking antidepressant medication. And if I've done my math right, that is about 1 out of 8 people are taking depressants. Females are 2 times more likely to take antidepressants than are males. And alarmingly, greater than 250 million prescriptions for antidepressants are written per year in the United States. It's hard to believe this number, and in fact, most prescriptions of antidepressants are not written by a psychiatrist, but by a family physician doctor, internal medicine doctor, etc. So my project focuses on a specific type of population who are depressed, which is patients who fail to respond to 2 or more courses of antidepressants. They're referred to as treatment-resistant depression. So there is about 40 different antidepressant drugs available worldwide in the market currently, or unfortunately, about 5 million of these depressed patients just in the United States are refractory to the treatment, despite taking multiple drugs or drug combinations. And less than 50% of the patients have a reduction or disappearance of the symptoms of depression after using antidepressant drugs. So there is no definite reason for why some patients have treatment-resistant depression yet. However, a common theory that limits the efficiency of current medication is that common antidepressants may be hitting the wrong targets in the biological systems of patients who are treatment-resistant. So when it comes to the pathophysiology of MDD, it has not been clearly defined, but we know that it begins with chronic psychological stress. And stress does damaging effects to not only to your brain, but to your whole body. And we know that stress increases inflammation by activating the resident microglia cells. The microglia cells are immune cells in our brain. They're found everywhere throughout our brain. And their role is to remove damaged neurons or to eat up damaged neurons. They can also protect against infections. And they're just important for maintaining the overall health of our central nervous system. And so recent advances in biological psychiatry has found that microglia cells play a major role in brain inflammation by releasing inflammatory molecules. These molecules, as you can see, are called interleukin-1 beta, for example, tumor necrosis factor alpha, just to name a few. So these pro-inflammatory cytokines increase the activity or increase the enzyme activity of IDO. IDO reduces the production of serotonin. And most of us are aware that serotonin is the key hormone that stabilizes our mood, feelings of well-being, and happiness. So this goes back to the monoamine hypothesis that I've talked about. And therefore, the depletion of serotonin finally leads to depressive symptoms. The pro-inflammatory molecules can also contribute to the hyperactivation of the HPA axis. This axis is actually our central stress response system. Once this is hyperactivated, it will eventually lead to an upregulation or to an increased concentration of cortisol in our body. And cortisol throughout our body, highly prolonged cortisol in our body can actually weaken our immune system, can lead to heart disease, can increase our blood pressure or blood sugar, and in this case, it can also lead to depression and neurodegeneration. So my project actually stems from our recent discoveries in our lab. Previously, we have found in MDD brain donors, we have found elevated oxidative stress and damage. We have also found shortened telomere length as a result of the oxidative stress. And what's intriguing, we also found elevated levels of this gene expression called PARP1. PARP1, we found it was interesting that there was elevated levels in depression individuals because of its various tasks. PARP1 has many tasks to do. It can regulate cell division, it can repair DNA, and it could also activate inflammatory molecules, which lead to inflammation and then eventually lead to, as you can see over here, cancer survival, metastasis, and other lethal effects in our body. And so PARP1 is highly activated in two events. The first event it can be activated is by a cellular process that can promote inflammation. A second event it can also be highly activated is by a DNA strand breaks as a result of the oxidative attack on DNA bases. So we figured that PARP1 is somewhat related or linked to inflammation. And so after discovering elevated PARP1 gene expression in major depressive disorder, we have found that PARP inhibitors like 3-aminobenzamide actually exhibit antidepressant-like effects in rats. We measured the effects of PARP inhibitor in rats, where rats were actually exposed to chronic unpredictable stress and social defeat, mimicking treatment-resistant depression in humans. And we found that PARP inhibitor actually produces antidepressant-like effects. And so from there, we wanted to focus on microglia cells because we think that they might be a mediator for inflammation, which eventually would cause depression. And also, in fact, recent studies have shown that microglia cells play a major role in various psychiatric diseases such as depression and schizophrenia. And therefore, they could also be a contributing cell to promote suicidal thoughts. So we speculated that once microglia are activated by stress, they release inflammatory cytokines, and this would cause depression. We wanted to put a PARP inhibitor on this first step. This would actually, if we put a PARP inhibitor, we speculated that this would suppress microglial activation, which eventually the suppression of brain inflammation would inhibit the depressive symptoms in rat models. And in fact, PARP inhibition PARP inhibitors have been used as a therapeutic drug for many diseases that have comorbid pro-inflammatory activation, such as cancer. It's very lucrative in cancer, cardiovascular diseases, diabetes, and arthritis. However, there is no study demonstrating that PARP inhibitors as a potential treatment for major depressive disorder. So our hypothesis was that we wanted to see how does PARP inhibitor work? Does it interfere with the inflammatory process? Will it suppress microglia activation? Will it upregulate it? What will it do? And so we expected to see that the suppression of microglia activation when treated with the PARP inhibitor. We also wanted to see the morphological effects or the shape changes of microglia when they were induced by chronic unpredictable stress. So again, we used a treatment-resistant model in rats. So we had 4 treatment groups. Our first treatment group was a control, so it wasn't exposed to any stress. Our second treatment group was the rats were daily injected with LPS, which produces an acute inflammatory response, and they were also exposed to unconditional stress. Our third treatment group was that we had the TR model, but we also injected the rats daily with fluoxetine, which you might know by Prozac. Fluoxetine is a protypical antidepressant currently in the market right now. The fourth treatment group, instead of injecting it with fluoxetine, we injected it with the PARP inhibitor 3-aminobenzamide. So after 30 days, the rats were euthanized, and we reached our specific, the first specific aim that we have is that we wanted to determine if PARP inhibition affects microglia activation in the rat hippocampus and the cortical white matter. So we're focusing on the hippocampal region in the rat brain. And so after euthanizing the rats, we washed them with PBS, blocked them. We used a primary antibody called anti-IBA1, which is a marker for microglia. The next day, we applied a secondary antibody. We mounted the tissues on slides with DAPI. DAPI is a fluorescent stain, so it lets us see the microglia cells once we look at them on the computer. And then they were ready to be taken with the confocal microscopy and then eventually for analysis. So we used the the images of the fluorescent sections were captured using the EVOS fluorescent microscope. As you can see, here is the IBA1 image and the DAPI image. And it was very stunning to see when you look at the microscope, it's stunning to see that there's no place in our brain that doesn't have a process of a microglia. So they were everywhere. And after capturing the images, they were ready for analysis. So we used the MCID analysis software, which is a very highly controlled software. We would take the region of the consideration that we wanted. We would circle the region that we want, and the computer would give you the percent of the area that is targeted. So it's very, very carefully controlled. So again, here's our first results with our 2-dimensional analysis. Unfortunately, we didn't find any significant differences in all 4 treatment groups. However, we did find the same trend in all specific regions of the brain. We did find the IBA1 immunoreactivity to be reduced by the PARP inhibitor and fluoxetine in both white matter and cortex. Again, same trends that we see in the CA1 hippocampus and dentate gyrus. We see that the IBA1 immunoreactivity was reduced in both fluoxetine and PARP inhibitor as well. Next, we wanted to examine the morphology of microglia cells in the rats treated with PARP inhibitors. So we used an Imaris software that this shows you kind of what the 3-dimensional analysis of the microglia and the processes. Very, very interesting software as well. Here's just a short clip that shows you the microglia and the processes and how we analyzed each one of them. And so our results, unfortunately, as you can see over here, when we measured the soma of the microglia cell, there were no morphological changes induced by stress. However, when we wanted to measure the processes, which are called the filaments or the dendrites in the microglia, we did find a trend in the second treatment group with LPS and stress. It trended towards longer and larger microglia processes. However, again, no significant differences in each. So this reaches us into our conclusions. We didn't find the morphological indices of microglia activation. They were not confirmed with the three-dimensional analysis, meaning that probably microglia was not activated since it should have shown in our 3D data. The data maybe not what we were hoping for, but it's very informative to knowledgeable adjustment to future research strategy. So in the future, we were thinking that, we wanted to determine maybe if microglia is actually releasing the cytokines. Maybe we should use another marker protein instead of IBA1. Or maybe we should use another PARP inhibitor that has better properties to pass into the brain instead of 3-aminobenzamide. And so also another future idea, we wanted to see if we would produce the same results, but if we would do this in the human hippocampus brain and see what would this look like. So thank you. This concludes my presentation.